In this video, we're going to cover translation. We're going to break down how genetic information flows from mRNA to protein. Now, if you haven't seen the previous lecture on DNA transcription, I highly suggest watching that first because it connects with this one. If you have seen it, let's do a quick recap to kickstart the lecture. Okay, so when a particular protein is needed by the cell, the nucleotide sequence of a gene is first copied into RNA. The RNA copies are then used to direct protein synthesis. This process is called gene expression. There are two major steps in gene expression: transcription and translation. Transcription is the copying of DNA into RNA. Let's go through this. DNA transcription begins with unwinding the DNA double helix, and one strand will serve as a template for RNA synthesis. This is done by RNA polymerase, and just like DNA polymerase in DNA replication, RNA polymerase can only assemble polynucleotides in the 5' to 3' direction. Okay? In bacteria, they have a single type of RNA polymerase. In eukaryotes, there are 3 types of RNA polymerase, with RNA polymerase II being the one responsible for pre-mRNA synthesis for all protein-coding genes. So to start transcription, RNA polymerase must be able to recognize the start of a gene known as the promoter. Now, recall the terms used to refer to the direction of transcription, which is downstream, and the other direction, which is upstream. The promoter sequence is said to be upstream from the starting site of transcription, and the segment of DNA being transcribed or copied is called a transcription unit or RNA transcript. Now, we can break down transcription into 3 stages: initiation, elongation, and termination. With initiation, RNA polymerase binds to the promoter in a precise location and orientation. And because it can only synthesize in the 5' to 3' direction, it must use the DNA strand that is oriented in the 3' to 5' direction as its template. Okay? RNA polymerase also requires transcription factors to help guide the binding of RNA polymerase, and this whole complex is called a transcription initiation complex. We go through this in detail in the previous lecture. The next stage is elongation. RNA polymerase will move along the DNA, adding nucleotides to the 3' end of the growing molecule. Then the last stage is termination. In eukaryotes, RNA polymerase transcribes a sequence called the polyadenylation signal sequence, which specifies a polyadenylation signal in the pre-mRNA. Okay, these 6 RNA nucleotides. And once these 6 RNA nucleotides appear, they're bound by certain proteins in the nucleus. And then about 10 to 35 nucleotides downstream from the signal, these proteins cut the RNA transcript free from the polymerase enzyme, and we release the pre-mRNA. The pre-mRNA will then undergo processing before it can be exported to the cytosol, which includes capping, splicing, and polyadenylation. We went through these steps in the previous lecture, okay? After these modifications, the mRNA molecule is ready for translation. Before these steps are completed, the RNA transcript is known as a pre-mRNA or precursor mRNA. Only correctly processed mRNAs are exported to the cytosol, where the information from RNA is translated into protein. So that's the first stage of gene expression, transcription. Let's move on. If you recall, in the DNA structure and function lecture, we covered how the structure of DNA carries information for specifying an organism. And we can consider each base, adenine, cytosine, guanine, or thymine, a letter in a 4-letter alphabet that's used to spell out biological messages. Just like different languages, organisms differ from one another because their DNA molecules have different nucleotide sequences. And focusing on RNA here, DNA and RNA are chemically and structurally similar, which is why DNA can act as a template for the synthesis of RNA. So the language and form of the message are quite similar. But when it comes to converting the information from RNA into protein, we're switching languages and using different symbols. Because there are still only 4 nucleotide bases in mRNA, but 20 different types of amino acids in a protein. And this is where the genetic code comes in, which is the set of rules by which the nucleotide sequence of a gene is translated into the amino acid sequence of a protein. Let's break this down. In the early 1960s, it was discovered that the sequence of nucleotides in an mRNA molecule is read consecutively in groups of 3. And because RNA is made up of 4 different bases, there are 64 possible combinations of 3 nucleotides. The genetic code is redundant. That is, several different codons can specify a single amino acid. So the genetic instructions for a protein are written in the DNA as a series of non-overlapping 3 nucleotide words. And these 3 consecutive nucleotides, or mRNA nucleotide triplets, are called codons. And each codon specifies 1 amino acid. Beautiful. Let's go through an example. We have our DNA molecule here. The 3' to 5' strand is the template strand because recall we, or RNA polymerase, can only synthesize in the 5' to 3' direction, which is why the template strand needs to run from 3' to 5', okay? So the nucleotide triplet, CGA, along the DNA template strand, starting from the 3' end, provides a template for GCU in the mRNA molecule. And GCU is the codon for the amino acid alanine, okay? Codons are written in the 5' to 3' direction. And it's important to note that the term codon is also used for the DNA nucleotide triplets along the non-template strands. So during translation, the sequence of codons along an mRNA molecule is translated into a sequence of amino acids making up a polypeptide chain, okay? Let's slow down for a second. Let's go through the codon table for mRNA. Codons are written and read in the 5' to 3' direction. So here we have the first, second, and third bases. And the amino acid is given in both its 3-letter and 1-letter abbreviations. Now, by examining this table, we can see that the same amino acid tends to contain the same nucleotides at the first and second positions and varies at the third position. And there are 3 codons that don't specify any amino acid, but instead act as termination sites or stop codons, these here, which signal the end of the protein coding sequence in an mRNA. Let's go through this. So we have UAA, UAG, and UGA. The way you can easily remember these stop codons is by saying, for UAA, you are annoying. For UAG, you are gone. And for UGA, you go away. Okay? And there is one codon, AUG, which acts both as an initiation codon and a start signal for ribosomes to begin translation. And as the codon that specifies the amino acid methionine, okay? The way to remember AUG, or the start codon, is you can say, are you good to go? Okay? That's the start codon. Wow, this is absolutely beautiful. Let's now subtract complexity and go through the important molecular components, and then we'll build a polypeptide. So we've covered how translation converts the information from mRNA to protein through a series of codons along an mRNA molecule. Now, the codons do not directly recognize the amino acids they specify. It's not like base pairing where they bind directly to the amino acid. Rather, translation depends on adapter molecules that bind to a codon with one part of the adapter and to an amino acid with another, okay? We can call these adapters translators, and they consist of small RNA molecules known as transfer RNAs or tRNAs. So the function of a tRNA is to transfer an amino acid to a growing polypeptide in a ribosome. The cell has all 20 amino acids in the cytoplasm, and the ribosome adds each amino acid to the growing end of a polypeptide chain. And so what brings the amino acid to the ribosome? Transfer RNAs or tRNAs. Let's take a closer look at the structure of transfer RNAs. This big boy is very important. From an overview picture, tRNA has a specific amino acid at one end of its 3D structure, this right here, while at the other end is a nucleotide triplet that can base pair with a complementary codon on mRNA, so this here. A tRNA molecule consists of a single RNA strand, and its structure can form hydrogen bonds between different regions of the molecule, so it folds back on itself, forming a double helical structure. Specifically, there are 4 segments of the folded tRNA that are double helical. So if we look at a tRNA molecule, it looks like a clover leaf, which is pretty cool, right? The molecule twists and forms to form a compact L-shaped structure that is held together by additional hydrogen bonds. Alright, and we both have the 5' and 3' ends of the linear tRNA located near one end of the structure. So these 2 regions are very important in protein synthesis. At the 3' end, this region acts as the attachment site for an amino acid, and the other region forms the anticodon. This right here, this loop here, is a set of nucleotide triplets that base pairs to a specific mRNA codon. Okay, codon and anticodon. So each tRNA molecule picks up an amino acid in the cytosol and drops it off to a polypeptide chain at the ribosome. And once it leaves the ribosome, it can go pick up another of the same amino acid. It's important to note that anticodons are written in the 3' to 5' direction to align with codons written in the 5' to 3' direction. Okay, let's go through an example. Consider the mRNA codon UUC, which translates to the amino acid phenylalanine, but we'll call her Penny, okay? The tRNA that hydrogen bonds with this codon has AAG as its anticodon and carries Penny at the other end. As an mRNA molecule moves through a ribosome, Penny will be added to the polypeptide chain whenever the codon UUC is presented for translation. But the question is, how does each tRNA molecule recognize the one amino acid in 20? Because a tRNA that binds to an mRNA codon specifying a particular amino acid must carry the correct amino acid. It's very specific. So recognition of the correct amino acid depends on enzymes called amino-acyl tRNA synthetases, which covalently couple each amino acid to their tRNAs. Let's go through this. The active site of each of amino-acyl tRNA synthetase fits only a specific combination of amino acid and tRNA. That means there are 20 synthetases. One attaches alanine to all tRNAs that recognize codons for alanine. Another attaches phenylalanine, or Penny, to all tRNAs that recognize codons for Penny, and so on. Each synthetase enzyme recognizes its designated amino acid, as well as nucleotides in the anticodon loop, and it catalyzes the covalent attachment of the amino acid to its tRNA in a process driven by the hydrolysis of ATP. Our end result here is the tRNA is now charged with its amino acid and released by the synthetase. We can also refer to this as a charged tRNA, and it can now deliver its amino acid to a growing polypeptide chain on a ribosome. Now, before we move on with dropping the amino acid to the ribosome, there's another type of recognition, and that is the pairing of the tRNA anticodon with the appropriate mRNA codon. Earlier, we covered how the genetic code is redundant, and there are several different codons that can specify a single amino acid. This suggests that some tRNA molecules can base pair with more than one codon. That is, some tRNAs require precise base pairing only in the first 2 positions of the codon and are quite flexible in the third. The flexible base pairing at this codon position is called a wobble, or mismatch. This mismatch explains why multiple codons that specify the same amino acid differ at the 3rd position, but have the same base at the 1st and 2nd position. Okay, that's tRNA. Let's now move on to how the mRNA message is decoded on ribosomes, the second important molecular component. Ribosomes are what allows the accurate and rapid translation of mRNA into protein. So it grabs onto an mRNA, captures and correctly positions tRNA molecules, and then covalently links the amino acids they carry to form a polypeptide chain. Beautiful machines. A ribosome consists of a large subunit and a small subunit, each made up of proteins and one or more ribosomal RNAs, or rRNAs. So this is the large subunit and the small subunit. Eukaryotic and prokaryotic ribosomes are very similar in structure and function. In both bacteria and eukaryotes, a large subunit and a small subunit join to form a functional ribosome, only when attached to an mRNA molecule. Now, the structure of a ribosome reflects its function of bringing an mRNA molecule together with tRNAs carrying amino acids. The small ribosomal unit matches the tRNAs to the codons of the mRNA, while the large subunit catalyzes the formation of the peptide bond that covalently links the amino acids together into a polypeptide chain. These two subunits come together on an mRNA molecule near its 5' end to start the synthesis of a protein. As the mRNA moves forward in a 5' to 3' direction, the ribosome translates its nucleotide sequence into an amino acid sequence, one codon at a time, using the tRNAs as adapters. Okay, so we're bringing in the tRNAs. The mRNA itself has a binding site for the ribosome. The ribosome also has a binding site for mRNA, as well as 3 binding sites for tRNA. So let's break down how the ribosome operates in the process of translation. Okay, so like I mentioned before, the ribosome contains 3 binding sites for tRNA molecules, called the A site, the P site, and the E site. So to add an amino acid to a growing peptide chain, a charged tRNA enters the A site, or the amino acyl tRNA binding site, by base pairing with a complementary codon on the mRNA molecule. The A site holds the tRNA carrying the next amino acid to be added to the chain. Then the P site, or peptidyl tRNA binding site, holds the tRNA carrying the growing polypeptide chain. Then the large ribosomal subunit shifts forward, moving the discharged tRNA to the E site before ejecting it. So the discharged tRNAs leave the ribosome from the E site, or exit site. Now, rather than trying to remember which site is which, understand that each site serves a purpose, and that is the A site is the amino acyl tRNA binding site, which holds the tRNA carrying the next amino acid. So A site equals the next amino acid. Then the P site is the peptidyl tRNA binding site, which holds the tRNA carrying the growing polypeptide chain. So P site equals polypeptide chain, and the E site is the exit. Okay, and this cycle of reactions is repeated each time an amino acid is added to the polypeptide chain, with a new protein growing from its amino to its carboxyl end until a stop codon in the mRNA is encountered and the polypeptide chain is released. Now, it's important to mention that rRNAs, not the ribosomal proteins, are responsible for the ribosome's overall structure and function. Ribosomal proteins help fold and stabilize the RNA core, so the rRNAs form the tRNA binding sites and act as the catalyst of peptide bond formation. Recall from the previous lecture that RNA molecules that possess catalytic activity are called ribosomes. Okay, let's now take everything we've covered and build a polypeptide. Similar to DNA transcription, we can divide translation into 3 stages: initiation, elongation, and termination. And all 3 stages require protein factors, and some reactions require energy, provided by the hydrolysis of GTP, or guanosine triphosphate. Let's start with initiation. At this stage, we bring together an mRNA, a charged tRNA, the first amino acid of the polypeptide, and the 2 subunits of ribosome. First, a small ribosomal subunit binds to both the mRNA and a specific initiator tRNA, which carries the amino acid methionine. Newly made proteins all have methionine as the first amino acid at their N-terminal end. In eukaryotes, an initiator tRNA carrying methionine is first loaded into the P site, okay, for the small ribosomal subunit, along with additional proteins called translation initiation factors. These factors are required to bring all these components together. Then, the small subunit loaded with the initiator tRNA binds to the 5' end of an mRNA molecule, which binds to the 5' cap of the mRNA. And then it's going to move downstream, okay, along the mRNA in the 5' to 3' direction until it reaches the AUG start codon. When the start codon AUG is recognized by the initiator tRNA, several of the initiation factors dissociate from the small subunit to make way for the large subunit, the large ribosomal subunit, to bind and complete the entire ribosome, okay? And there's a name for this. There's a name for when the mRNA, initiator tRNA, the small ribosomal subunit, and the large subunit come together. This is called the translation initiation complex, all right? Now, the cell also expands energy obtained by hydrolysis of a GTP molecule to form the initiation complex. Now, recall that the initiator tRNA is bound to the P site, so the available A site is ready for the next amino acyl tRNA, okay? Protein synthesis is ready to begin with the addition of the next charged tRNA to the A site. A polypeptide is always synthesized in one direction, from the initial methionine at the amino end, also called the N-terminus, all the way to the final amino acid at the carboxyl end, also called the C-terminus. So that's initiation. The next stage is elongation of the polypeptide chain. In the elongation stage, amino acids are added one by one to the previous amino acid at the C-terminus of the growing chain. Each addition involves several proteins called elongation factors, and it occurs in a 3-step cycle. Let's break this down. The first step is codon recognition, which we touched on earlier. The anticodon of an incoming amino acyl tRNA base pairs with the complementary mRNA codon in the A site. Recall that only the tRNA with the correct anticodon will bind and allow this process to continue. And in this step, we need GTP. The second step is peptide bond formation. A tRNA molecule of the large ribosomal subunit catalyzes the formation of a peptide bond between the carboxyl end of the growing polypeptide in the P site and the amino group of the new amino acid in the A site, okay? This right here. This step removes the polypeptide from the tRNA in the P site, okay? And it's going to attach it to the amino acid on the tRNA in the A site, okay? This right here. That's the second step. The third step is translocation. The ribosome translocates the tRNA from the A site to the P site. At the same time, the empty tRNA at the P site is moved to the E site where it is released. The mRNA then moves along with its bound tRNAs, bringing the next codon to be translated into the A site, and the cycle repeats again. And we're going to be using GTP to power these reactions. So that's elongation. The final stage of translation is termination. So elongation is going to continue until a stop codon in the mRNA reaches the A site. The nucleotide-based triplets UAG, UAA, and UGA acts as signals to stop translation. A release factor, okay, a protein shaped like an amino acyl tRNA, this one here, binds directly to the stop codon in the A site. All right? The release factor causes the addition of a water molecule instead of an amino acid to the polypeptide chain. And this reaction is going to break the bond between the completed polypeptide and the tRNA in the P site, releasing the polypeptide through the exit tunnel. Then at this point, the ribosome also releases the mRNA and dissociates into its 2 separate subunits, which can then assemble onto another mRNA molecule to begin a new round of protein synthesis. And this whole process starts all over again. In this lecture, we learned the second stage of gene expression, We covered the genetic code and how the sequence of nucleotides in an mRNA molecule is read consecutively in groups of 3. We broke down the functions of tRNAs and ribosomes and how we can break down translation in 3 stages: initiation, elongation, and termination. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist. Hey, stop procrastinating!